started for today. Does anyone have any questions about anything you covered last week? And any issues yet on the homework assignment? Okay, then I guess we can get going with this week's material. So what we're going to be looking at this week is the topic of gene and protein expression. So this basically refers to activities of genes or proteins within an organism. And this is one of the major kinds of data that has really made it possible to start thinking about what are called systems biology approaches. So to start thinking about how organisms or cells or tissues function as a coherent system of many genes rather than thinking of them in terms of individual components. So what we mean by expression basically flows from the concept of the central dogma of molecular biology, which we've discussed a number of times at this point. We have DNA. DNA is transcribed into RNA. And often when we're talking about expression, we're talking about RNA. So expression would mean how much RNA is present of a particular kind of gene. Often we're interested literally of how many transcripts are present from a particular gene in a particular cell type, in a particular tissue at a particular time. And we may be interested in a finer level of granularity, such as what splice forms, which particular exons, and so forth are present at a particular point in time. Usually we would assume that the functional element within the organism is actually the protein rather than the RNA. So there are reasons why we generally look at RNA rather than protein that I hope will become apparent later, but sometimes we do refer to protein when we're talking about expression. We might be interested in knowing how much of a protein is present in a particular cell, or maybe even a particular location in a cell at a given point in time. We might want to know if there are different splice isoforms of the gene that lead to different amino acid sequences, which of those are present. And very often with proteins, we would have additional information important to us, such as which post-translational modifications. So it may be that we're looking at a protein whose activity is different depending on whether it is phosphorylated or not phosphorylated, and we would often be very interested in quantifying those particular differences. With proteins, we often have a finer granularity of location of expression. So with RNAs or proteins, we'd often worry about expression at the level of tissues or cell types. With proteins, we're often looking at an even finer scale, so it may matter whether the protein is present in the nucleus versus the cytoplasm, and that's a kind of activity or a kind of localization that is harder to get for RNA, and often less relevant for RNA, but often available for proteins. So that basically is what I mean by expression. Are there any questions about any of this yet? So there are a lot of things we would want to know about expression. At the simplest level, of course, we want to know which genes or proteins are expressed. And often we want to know where they're expressed. So I've mentioned a little bit of this already. We might want to know which cell types they're expressed in, which tissues, maybe which subcellular localizations. Very often we're studying expression because we care about when proteins or genes are expressed. So in some cases, that refers to temporal expression. So at different points in time, different proteins will be expressed. If we're studying development, for example, a very important question in trying to understand how development of organisms works has to do with developmental stages at which different genes become active or inactive. And that's one of the main things you would be looking at if you're studying how an organism develops. Which modules of genes are turning on or off as you go through the stages of development. There can be multiple different time scales at which we worry about expression. So if you are studying the cell cycle, so you're studying cell division on the level of individual cells, then we might be interested in how expression changes over the course of the cell cycle, which might be a time scale of hours to days, depending on the cell type you're looking at. And there can be both finer and coarser grains of time scale in which we are interested in temporal expression. We're also very often interested in condition-specific expression. So if you are studying, let's say, basic research into metabolism, you might care about how expression changes when organisms are provided different nutrients or different other sources of key environmental feedback that they need available to them. And you might be interested in which genes turn on or off depending on those kinds of environmental conditions. We might be interested in responses to various kinds of stress. So in let's say a study of yeast, for example, we might want to know what happens to expression if you expose your yeast to heat or cold or any other kind of stress. And typically what you would find is that there are modules of genes that will turn on specifically in response to cold, let's say. There are other modules that will turn on in response to almost any kind of stress, and of course modules that will turn off under various kinds of conditions. And a lot of work in these sorts of systems approaches to biology has to do with these questions of figuring out what these modules are and what they're doing. Finally, we are often interested in aberrant expression. So we might be interested in studying a disease condition, and we want to know how gene activity is changing in response to the disease condition. So if we're studying, let's say, cancer, we might be interested in which modules of genes are turning on or off as the tumor develops and as it goes through different stages of progression. So which genes turn on when the tumor first develops, which genes turn on or off as the tumor enters metastasis, and so forth. So that would be one example of this. And we're also often interested in how expression changes in response to mutation. So this may be a disease mutation, it may be just a natural variation of the kind you guys learned about last week. So one of the things that has interested a lot of people in the field in trying to understand how genetic variations influence phenotype is discovering what are called eQTLs, or expression QTLs. Does anyone remember what a QTL is? Yeah, QTL is a quantitative trait locus. It's a genetic variation that affects something that is measurable with a real number, so height or weight or so forth. And there's a lot of interest in discovering these QTLs where the real number they modulate is activity of genes. And this is something a lot of people in the field are looking at as a way that natural genetic variations in a species might have subtle effects on various phenotypes. So maybe having a T versus a C at a particular site in the promoter region or a particular gene causes your activity level of that gene to be higher than someone who has the other variant. So that's something a lot of people are looking at in the field. So any questions about any of this yet? All right, so normally when we study expression, we have to think very carefully about experimental design. So what kind of data are you looking at? Usually you're not going to have a single sample that you look at. It's often not going to be meaningful to look at one organism and one condition at one point in time and say anything about expression. Really, you need to look at expression as a relative thing, something that changes between different samples. So you usually will be looking at a set of samples. If you're studying developmental biology, maybe you're looking at a time course of samples. You might be looking at how gene expression changes in a particular tissue, in Drosophila, over 10 days of Drosophila development, or whatever you're looking at. Maybe 10 is not the right number for Drosophila, but maybe that would make more sense for, say, mouse embryos. If we're looking at a medical condition, often we would be interested in variation among patients or between patients and controls. If you were looking at gene expression in tumors, for example, maybe you're interested in how gene expression varies between 10 different patients who have what look like clinically similar tumors. And by studying how patterns of expression are different among these people, you can discover what are called different subtypes of tumors that may respond to different kinds of treatment or have different prognoses. In more basic research contexts, you may be studying variation among different perturbations. We might try a set of different carbon sources, for example, for growing our bacteria or yeast or whatever, and then we might be interested in how expression programs change as we grow our organisms on different kinds of media. So all of these would be examples of experiments we might set up, and as we'll see when we get to the next class and look into some of the computation and statistics that goes into this, it can be quite important how you set these up and how you think about how many samples you have, how many replicates for each condition, and so forth. So that basically is the sort of thing we would study with expression. But what I want to spend most of this lecture on is the question of how we measure expression. So as with many of our prior modules that we've covered in this class, I need to spend a bit of time talking about the biology, and really more specifically the biotechnology, of how you actually measure expression. Because as we'll see, different kinds of technologies have different trade-offs involved with them. And when you're doing analyses that really depend very heavily on the kind of data you have available, you need to know something about these trade-offs and what options you have and why one or another might be appropriate for different conditions. So I wanted to start by talking about RNA expression measurements. And as I mentioned before, we usually are going to look at RNA expression. Usually when we're talking about expression and we're looking at large data on expression, what we're measuring is RNA. Now, even though the protein is usually the functional thing, that's what we usually care about, RNA is usually what we have. And the reason really has to do very much with the technologies. There are simply much better technologies for measuring activity of large numbers of genes than there are for measuring activity of large numbers of proteins. And that will become apparent as we look through these technologies. And this is just a few. This is not an exhaustive list. It's just a few I wanted to mention, either because they are important today as things you might be using, or they're historically important and will help you understand the kinds of data resources available. And so this is just a partial list that I want to go through today, spending more time on some than on others. So any questions before we get to this? All right, so I will start with real-time PCR, RTPCR. You'll sometimes also see the term qPCR, quantitative PCR. It's all basically the same thing, measuring amounts of RNA using a variant of PCR. So we've seen PCR before. You should all be familiar with what PCR is. It's basically a way of amplifying a typically small amount of nucleic acid using a pair of primers to pick out a region of interest, and then repeatedly amplifying it, approximately doubling the number of strands in each round of amplification until we have a large quantity. Traditionally, PCR is just used to go from a small quantity to a large quantity without paying too much attention to exactly how much you have. But where RT-PCR comes in is by using the fact that you can actually fairly precisely quantitate how much amplification you're doing in each step. So if you, for example, have a very small amount of RNA, probably you're not going to be able to accurately measure that. It's hard to accurately measure a small quantity of something. It's much easier to get an accurate measurement of a large quantity. And the idea behind RT-PCR is simply that we can look at our small quantity of RNA we care about. We can see if it's measurable accurately, and probably it isn't at first. But if it isn't, then we can run a round of PCR, and a round of PCR will come very close to doubling the amount of RNA we have. So we can then see, once we've doubled the amount, do we now have enough to measure it accurately? If not, we can do PCR again, and we now know we've got 4 times as much as we started with, and we can see if that's measurable. If not, we can do it again. We have 8 times as much as we started with, see if that's measurable. And essentially, you just keep doing this until you have enough that you can accurately measure how much is there, and then you just need to work backwards, see how many times you doubled it, and use that to adjust the amount you measure to get back how much was originally present. That's basically the idea behind RT-PCR, and it tends to be a very good method for accurately quantifying amounts of RNA. So if you really need a very accurate measurement of the amount of a particular gene, a particular sample, this tends to be a good technology for doing that. So any questions about that? All right, so that is really the big advantage of RT-PCR, but it has some appreciable disadvantages. And the main one is that it is not what we would call a scalable technology. It's relatively easy to do this for 1 gene or 5 genes or 10 genes. It's not something you want to use if you were trying to do this for 10,000 genes. So it's labor intensive. Generally, you're not going to be able to easily automate every step of this process, and you're going to need a primer pair for everything you're trying to measure with this, and that's usually not something you want to generate for thousands of genes. So this is typically going to be our best choice if you're looking at a few genes, but we're often interested in cases where we're trying to profile every gene in an organism. This is not generally going to be the technology you want to use for that. An alternative to that, which was really the first large-scale way of looking at gene expression, was an approach that's not really used anymore called EST mapping. And I say it's not really used anymore, although it's very closely related to what many people are doing today, which I'll get to later. But I guess I should ask, can anyone tell me what an EST is? I have used that term before in this class. So an EST, it stands for Expressed Sequence Tag. It's essentially just a raw sequence read off of a sequencing machine, basically what we would think of as a randomly sequenced piece of RNA. So you take all the RNA in the sample you're interested in, so maybe you're trying to study expression in human livers, so you gather lots of livers, you purify them, get out your RNA, and just randomly sequence whatever RNA is present. That gives you ESTs. And the way you use that as an expression technology is you simply take the sequence information coming off of your sequencing machines, blast it against your genome, see where it aligns, and anywhere you get ESTs lining up to a gene, you keep track of the count of these, normalize it by the length of the gene, and that gives you an approximate expression level for that gene. So that tended to be a good technology for its time. It gave us a way of using the early sequencing technologies, the Sanger sequencing, to get a rough measure of how active genes were across an entire genome. So you could take these technologies that were already available. There are lots of these ESTs already mapped to the genome of any major organism you're looking at. So if you want to know at a very rough level how active different genes are in the human, you can go to a human map viewer, human genome browser, look at the EST alignment tracks, and you can get a rough idea from how many ESTs are aligning at any point in the genome, how active the expression is. Now that's very approximate. It needs to be done separately for any condition or any tissue type or whatever that you want to look at. And because you don't get that many reads aligning to any one position with these older technologies, there simply isn't enough sequence, you're getting a very imprecise measure. Most genes, you might have 0 or 1 ESTs. You can't really tell if that is a mistake or a low activity level or very low versus slightly low. You might get maybe 10 or 20 aligning to a particular position, which means that you might have a measurement that's accurate to maybe the order of 10 or 20% error, which is still not great. So this is something that was nice for the time. It gave you a way of discovering activity genome-wide, but it's not really used much anymore. It's still worth being aware of because you will see it in the databases, though. One related technique I wanted to mention is another thing that's not really used anymore called serial analysis of gene expression, or SAGE, that I mentioned, again, in part because you'll still see this term sometimes in the data resources you look at, but in part because I think it's a really interesting application of molecular biology to try to improve the way these techniques work. And the idea behind SAGE was basically to more or less do EST mapping, but to use some clever molecular biology to get more out of your EST mapping. The basic idea being that you could take your RNA, convert it to what's called cDNA, so copy DNA, which basically just means you convert the RNA to the equivalent DNA, and then you apply restriction enzymes to your DNA. Does anyone remember what a restriction enzyme is? Yeah. Chosen the right restriction enzymes, you can get these kinds of patterns of overhanging edges between your strands. And you can then get your strands to kind of randomly piece themselves back together. They'll all have the same overhanging ends, so even though you might have had a few different genes that originally gave rise to these, they can reassort into a completely different set of hybrid sort of pseudo-transcripts, each of which might be made up of, let's say, 20 or so of these little pieces left after the restriction fragmentation. You can then apply a ligase, which is a kind of enzyme that will glue these double strands back together. And then instead of sequencing your original DNA, you sequence these hybrid strands. So can anyone tell me what you get out of that? What's the point of doing that?
It seems like it's a lot less sequence.
Yeah, basically you're getting a lot more out of each sequencing reaction because now each of these has measurements effectively of activity of 20 or so genes instead of activity of a single gene. So this was something really useful back in the Sanger sequencing days when sequencing was expensive and you couldn't get as much sequence as you wanted. It's not used so much now that we have better sequencers and you can just get a lot more reads, but there's no reason in principle that you couldn't keep using this technology. So in a way, like ESTs, it was something very suitable to its time. It was a good way of getting more out of the technologies available to us. And there's no reason we couldn't keep using this. There's no reason we couldn't use it to get much more accurate expression measurements with the newer technologies. It's not used so much anymore, but I mention it in part because, as I say, it's still in the databases, in part because I think it's an interesting thing to be aware of, nice way you can think about how molecular biology can improve your computation, and in part because I'm not convinced that this kind of technique isn't going to be useful again. So that's sage tagging, though. It's not too important as something you're likely to do today, though, so I don't want to dwell on this. Are there any questions about that? All right, so the next technology, and the thing that really made expression studies possible on very large scales, were microarrays. And I believe you've heard about microarrays once before in a different context. So microarrays are an important technology for studying genetic variation, so often SNPs are typed with SNP arrays. They're actually much more important as a technology for studying gene expression. So the way a microarray would work to study gene expression is that we would have a sample of nucleic acid, and we would attach fluorescent probes to our sample, fragmented into small pieces, and then we would have an array covered with spots where each spot would have many copies of a particular DNA strand, and we would try to figure out what strands were present in our sample by using the fact that they should anneal to complementary strands on the microarray. Because our sample has been fluorescently labeled, by looking at the intensity of these fluorescent labels, and therefore the intensity of the spots that things have annealed to, we can figure out how much RNA in our sample was complementary to any particular spot, and thus, in theory, if we've chosen our spots carefully, approximately how much nucleic acid there was present of any particular gene of interest to us. So we might get an array that has one spot for every known human gene, and then by looking at the intensities of these spots, we can figure out which genes are highly expressed, which genes have low expression, and so forth in a particular sample. And this was really the technology that made it relatively cheap and easy to look at activity of very large numbers of genes in relatively large numbers of samples. So to do a study where you could ask about expression of every gene in the human genome in 100 cancer patients, let's say. So any questions about that? All right, so it is worth knowing that usually you're not going to be able to get absolute expression levels from this technology. We would always talk about expression relative to another sample. So typically the way a study designed for this would work is that we will have a set of samples of interest to us, and we would run a set of arrays for those samples, but we would also have a control sample. And we'll always be talking about expression of a particular spot in one sample relative to the control. So the simplest thing to do, and what's often done, is to use simply a pool of all of your samples to get a kind of average expression level as your control. In some contexts, there's some other natural control that would make sense. So if we're talking about the cancer example, maybe we're studying lung cancers, then it might make sense that the control you want is normal, healthy lung tissue, and then the samples would be various examples of cancerous lung tissue. but the kind of default thing is just to use an average of all of the individual samples. Can anyone think of why it might be that we would want to do this kind of relative expression? Why can't we just look at two different spots on the same array and say that one spot has bright color, another spot has dim color, and therefore the bright one corresponds to a high expression gene, the dim one a low expression gene? What's wrong with that?
If you have a longer strand of DNA, there's going to be more fluorescent tags, so if you less of a longer strand look brighter than more of a shorter.
Yeah, so basically different strands will have different propensities to anneal to one another. Might be that you have different lengths of strands, and then a longer one will have a more stable binding, so you'll get a brighter signal. If you have some strands that are more GC rich and others more AT rich, you get tighter binding in general with GC rich strands and AT rich ones. And strands often have some propensity to fold back on themselves, and if one strand is more prone to folding back and annealing within itself, that will lower its ability to anneal to a complementary copy. So basically what all of that contributes to is the fact that you cannot look at two different spots on the same array and say anything about relative expression of those spots based on their brightness. What you can do is look at the same spot on two different arrays and talk about relative activity of those two different spots. And that's why we'd always talk about these expression measurements in terms of relative expression between a sample and a control. Often that will be visualized by thinking about the sample being, let's say, labeled with red. So canonically there's a red dye used for the sample, a green dye used for the control. And then these will often be visualized by looking at the relative brightnesses of the red or green signals. So if we are seeing a red spot, that would tell us we're looking at something where the sample has relatively high expression. A green spot means the sample has relatively low expression relative to the control. And you really can't read anything into the absolute brightness of one spot versus another. So it doesn't really matter that this spot is bright yellow and this spot is maybe almost black, so very dim yellow. You can't read anything into that. All right, so any questions about any of that? Right, so when you look in the data sets, you'll often see something that looks like this image here. This is a computational visualization of a microarray study. And it's worth noting that this is not the actual microarray. You'll often see these when you look at microarray data, but what this is actually showing is a full study involving many different samples. And typically, each row running left to right would represent one probe on the microarray, so one spot, and very often that's going to correspond to a particular gene, and then each column running top to bottom would represent a sample. So in this particular case, each column represents a single cancer patient in this study, and each row would represent a spot, so typically a gene. So this is often a useful way of visualizing. We'll see that when we get into some of the analysis methods we'll look at next time, that having things represented in this form lets us kind of identify patterns of regularity. So we can see that maybe there is a group of patients here with more or less a common pattern of expression, and a group of genes that behaves in a common way across those patients. So that's why it's visualized this way. But be aware that this is not the actual microarray. This is just a way of looking at the data from the study. When you look at the raw data, it's worth knowing that usually this will be reported in the form of a log ratio of sample to control. So when you see the actual numbers in these, you'll typically see numbers more or less on the range of -5 to +5, something in that range. And the reason for that is that you're looking at the log of the ratio of sample to control. So if you have more activity in the sample than the control at a given spot, you'll have a number greater than 1, so the log will be positive. If the control has higher activity, you'll get a negative number because the ratio is less than 1, so the log is negative. So just be aware that that's usually how the raw data is stored. Any questions about any of that? All right, so these had a lot of advantages that made them really the standard technology for a long time. They were very high throughput. So So it's easy to automate the use of these microarrays. You could get a microarray, you can run that on a sample, you can take a fluorescent image of it, and basically at that point everything is automated. Once you've got your fluorescent image, and even actually getting to that, a lot of it's robotic automation. But once you've got your fluorescent image, you can put that through a computer. The computer can measure the intensities of the fluorescent probes, assign that to particular genes, come up with these raw data files, apply various other kinds of analyses to these data files. So that's a lot of what made this really practical. It was also a very cheap technology as these kinds of things go. Much cheaper at the time than doing sequencing. It's still cheaper than sequencing. And when people are talking of doing these kinds of things on large scales, that's not a small consideration. So I'm not sure what the latest prices are, but more or less you can go to a catalog and order a, let's say, a human gene microarray, and I think this would probably cost on the order of $100 to $200 at this point. As laboratory experiments go, that's pretty cheap. And that was a lot of what made this something that could put large-scale gene expression studies in the hands of basically any biology researcher who wanted to do them. They did have some considerable disadvantages, though, and one of them is that they are a noisy technology. You get much less accurate measurement with a microarray than you would get with, let's say, RTPCRs. And because of this, you typically need to worry a lot more about replicates, so you need to worry a lot more about having multiple samples if you want an accurate readout, and it becomes an issue when we get to computational analysis. So we'll see next time some of the kinds of analysis algorithms and statistical tests you do, and when we get into those, as we'll see in the next lecture, it becomes very important to worry about how noisy your data is, because the more noise you have, the harder it's going to be to get statistical significance, and you need to make up for that in other ways if you want a successful study. It's also a very sensitive platform, and sensitive in the sense that lots of different things can change your results on your experiments. So if you have prepared your samples two slightly different ways, you will see gene expression differences between the samples. So even though you may be looking at exactly the same sample, if it sat for a day between your tests, or you ran the experiment 2 times, you did your purification twice by hand, and you did it even slightly differently, that will show up in your measurements. If 2 different technicians ran your samples, you will see differences between the samples if you ran them on different days, and definitely if you use different microarrays. So you order one of your microarrays from one company, one from another company, that will show up as differences between your samples. And because you're very often looking for relative differences between different tests, this becomes an important thing to worry about. If you're trying to find the differences between some sets of samples, it may be that the differences between which technicians ran the samples overwhelm the differences that you're actually trying to look for, the real biological differences. So all of that goes into why these are difficult technologies to work with. And a lot of what goes into using these has to do with computational and statistical methods to try to deal with how noisy these are and get more robust readings from them. So that's just something to be aware of when we get into how we analyze these. Any questions about any of that? All right, so the next technology I wanted to mention is something called Nanostring. And Nanostring is a little unusual among the technologies I talk about in this class, because this is a proprietary technology. There's one particular company that sells the Nanostring device, and usually we don't worry too much at our level of abstraction. We're thinking of which company is making a particular technology, but in this case they have something unique enough and that's important enough for a particular niche within this field that I think it's worth knowing about, even though it's a proprietary technology. So the basic idea behind Nanostring is to do something that is, in some ways, a little like RT-PCR on a large scale. Essentially, the way the technique works is that you have pairs of strands of nucleic acids, sort of like primers, one of which is used to gather the RNA you're looking at and anneal it to a glass slide, sort of like a microarray, and another of which is used to label those strands of RNA. And this technique of having these pairs of strands allows you to get very high specificity, to pick out very precisely the strands you're interested in, and basically get rid of a lot of the noise of annealing a single kind of strand to a particular nucleic acid. And what all of that contributes to is that this is a technology that lets you get very high accuracy, so basically comparable to RT-PCR, maybe not necessarily exactly as good, but more or less on the same scale, while getting what we might call medium throughput. So you're not going to be able to look at 100,000 spots or 1 million spots like you can look at on a microarray, but you can look at hundreds of genes simultaneously. So this has become a very important and useful technology for getting very accurate measurements for large enough numbers of genes to start thinking in terms of complex biological systems. So to think of modules of interacting genes and look at these genes simultaneously. It is, as I say, a proprietary technology that creates a lot of disadvantages, and it's not quite a high throughput technology. You do need these pairs of nucleic acids, sort of like getting pairs of primers for RT-PCR for every strand you look at. So it's not something you can use whole genome, and it's not something you can use to measure activity of anything you weren't looking for already. So it, as I say, has an important niche it's worth knowing about because there are important ranges of experiments where this is the right technology, but it's not going to be necessarily a replacement for microarrays or other techniques that fill that niche. Any questions about that? All right, so that brings us to the technology that is really rapidly replacing everything else in the field, and that is next-generation sequencing. So we've talked about sequencing in a lot of different contexts already, and when we use this for RNA, it's commonly referred to as RNA-seq, and this is really becoming the dominant technology for looking at gene expression. Basically, this is the same thing as EST mapping, except that we're using the newer sequencing technologies where it becomes possible to get a very high number of strands. So you can talk about covering an entire genome at a 100x, so 100-fold coverage on average over the genome, and therefore getting pretty accurate quantitation. You can do whole exome sequencing at 1000x, so 1,000 strands on average covering any point in the genome. So this can give you quantitation that is not as good as Nanostring or RT-PCR, but much better than microarrays, and definitely much better than the old EST technologies. This also has the very big advantage, like EST mapping, that you don't need to know in advance what you're looking for. If you're interested in a particular gene, you will probably see expression of that gene if it's expressed, but you can also discover that something you didn't know was a gene, or you knew was a gene, but didn't know was active in a particular condition, is active there. You're basically getting a profile of an entire genome or an entire exome, whatever else you want to look at, and that can be a very appreciable, large advantage to these techniques. You can discover new splice forms, you can discover that there are differences in expression that you weren't aware of, you can discover non-coding genes that you may not have known were genes, and so forth. It still hasn't quite displaced the earlier technologies. So if you need really high accuracy, you would still use RT-PCR or Nanostring, and people still use microarrays for various situations, because even though the cost of sequencing is very rapidly dropping, it's still more expensive than microarrays, not by a ridiculous amount, but by maybe 3 to 5 times as expensive, and so when microarrays get the job done, often that would still be the thing you'd use. But I think microarrays are quickly becoming an obsolete technology, and RNA-seq is really becoming the standard thing you would use if you don't need very high accuracy. Any questions about any of that? All right, so that basically is RNA expression. And as I mentioned, usually when for looking at RNA expression for very large numbers of genes or even whole genome, we don't have anything equivalent for protein expression. So even though ideally what we would really want to see is the proteins, usually we get the RNA and we kind of pretend that the amount of RNA is a good proxy for the amount of protein. Those two things will be correlated. There's some debate in the field about how closely they're correlated, but basically RNA is what we can get, so usually that's what we would use. Still, it is worth knowing about technologies for protein expression, because in some cases this really is what you need and what you want to use. So I've divided these technologies into three basic groups. So immunological methods, which covers actually quite a lot of related technologies. Gel-based methods, typically 2D page, which I'll talk about in a few minutes. And mass spectrometry, so a technology we've seen before in other contexts. And these more or less are the technologies we would have available for looking at protein expression. When we talk about immunological methods, basically these are all methods that depend on the simple idea of raising an antibody to whatever you're trying to study, labeling the antibody, and then using the antibody to label your sample and quantifying how much of the protein was present based on how much labeled antibody you can see annealing to your sample. There are a lot of variations on this. Any of you who have been through any of your lab classes yet have probably done at least some of these. So Western blotting would be the simplest one. So that's just antibody labeling a standard polyacrylamide gel. That can give you very accurate quantification of single genes or a few genes. You can study fixed cells or tissues, what's called in situ hybridization. That can give you a way of quantifying activity with very fine localization to see how it differs between tissues or between cell types within a tissue, or even between subcellular localizations within single cells. Then there are more exotic variants, such as antibody arrays. So an antibody array is very much like a microarray, except that your spots are coated with antibodies to different proteins instead of with nucleic acid strands, and that can give you a way of profiling amounts or expression of different proteins on relatively large scales. So these techniques tend to give you very good quantitation. You can pretty accurately measure how much protein you have, and they give you kinds of information you can't get easily from nucleic acid, such as localization. But they do have some very significant disadvantages, and the biggest one is that you need an antibody for everything you're trying to measure, and it's not necessarily trivial to get an antibody. You're not going to be able to study an entire proteome, so all the proteins in an organism this way, and even if you're just interested in one protein, there's no guarantee you will be able to raise an antibody that works well for that protein. So this is something that has a pretty good chance of working for a small number of proteins, but there may be a significant amount of work involved if you're trying to study a new protein in this way, and it's not something you can use for looking at more than the order of maybe tens of proteins at a time. Because of that, it's not going to scale to the kinds of scales that we think of when we talk about RNA expression. Any questions at this point? So the next technology, which is a more high throughput technology, is 2D page, so 2D polyacrylamide gel electrophoresis. We've talked about standard page gels in various other contexts. So 2D page is basically a way of trying to do the same sorts of things we would do with a standard or 1D gel, but coming up with a second axis of variations, we can try to separate out larger numbers of proteins. And the way this works is that we first try to separate proteins by something called the isoelectric point. So the idea behind this is that we can set up a gradient of pHs, and a gradient of pHs is going to have the effect of that as proteins move through the gradient, they will change their charge. If you're in low pH, there are lots of free positive ions, so ionizable groups of the protein will tend to shift to their more positive state. So if you have a group that is, let's say, an acidic group, it's going to tend to be protonated in that state. Basic group will tend to be deprotonated. And the reverse will happen at high pH. And so if you put your proteins in this gradient and put them under an electric field, they will tend to move based on their charge. So if they're positively charged, they'll move towards a negative charge in your voltage potential. And they'll keep moving until they hit a pH at which the charge becomes net zero. So as a, let's say, a positively charged or negatively charged protein is moving towards low pH, its charge will keep getting closer and closer to 0, and when it hits 0, it just kind of stays where it is because it's no longer being acted on by the electric field there. So that point at which your protein loses its charge is known as the isoelectric point. And for the purposes of this technology, what matters is simply that different proteins have different isoelectric points, so you can separate out proteins. Different proteins will migrate to different regions within this pH gradient. You can then take your proteins at these different points and run them in a second dimension on a standard polyacrylamide gel to sort them by mass. And because mass and isoelectric point are more or less independent measures of a protein, more or less independent properties, what you hope is that your proteins will spread out nicely on your gel. So some proteins will have different isoelectric points, some will have different masses, and what you hope you get is lots of little spots where each spot is more or less a single kind of protein. That gives you a way of separating out maybe on the order of a few hundred proteins, and then by looking at how dark a spot is, you can get an idea of how much of a given protein is present. It's not necessarily easy to figure out what protein is in a given spot, but there are ways of predicting what the pi or the mass of a protein is, and there are ways of course, experimentally cutting out the spot and figuring out what's there. And you can also get other kinds of information, such as post-translational modifications. So there are characteristic shifts you would get from a protein being, say, phosphorylated that you can use to figure out these variations between different samples. Now, as is often the case, the idealized thing that I'm presenting in these kind of cartoon images is a lot cleaner than the real thing. So this is an image of a real 2D gel. You can see it's not nearly as ideal. You get a lot of nice spots, but also there are a lot of artifacts of the gel, spots running into one another, a lot of kind of messiness to it. So dealing with the real thing is not necessarily an easy problem. It's not nearly as easy to automate as dealing with sequencing, let's say. But that is the kind of data we actually get, the raw image data. There are ways to improve on this, and one I'll mention here is what's called DGE, or difference gel electrophoresis, which is basically a way to try to get around some of the difficulty of interpreting these gels by doing something very similar to what we would do with microarrays, and basically take a pair of samples, fluorescently label them with different labels, and then run them out on the same gel. That gets around some problems of having to figure out which spots on one gel correspond to which spots on another gel, which is not an easy thing to do because gels are not going to run exactly the same way every time, and to see more subtle effects that will let you figure out if there is a slight difference in how often a protein is phosphorylated in one sample or another, or slight differences in how much is present in one sample versus another. I mention this in part because it's a really useful technique for these things, and in part I'd like to mention it because this was invented at Carnegie Mellon. So this came from the Minden lab here at Carnegie Mellon, so I think it's a nice thing to be able to talk about. And this is a nice technology that gives us a way of profiling large numbers of proteins. This can resolve on the order of a few hundred proteins, give us information on modifications that we can't get from some of these other technologies, but it does have its disadvantages. It has a limited, what's called dynamic range, which means that you can't see a many orders of magnitude difference in different proteins. If you have a very high abundance protein, that kind of hides the low abundance proteins. That's something people are working on, but it's still an issue. It can be hard to resolve nearby spots, and it can be hard to automate and hard to compare among different gels. A lot of people are working with mass spectrometry as an alternative to this. So mass spec we've seen before. It's a way of measuring masses of different molecules in a solution. And this is commonly used in the form of tandem mass spectrometry, or MSMS, where we would break a protein down with a high-level fragmentation into peptides, so maybe 10 or so amino acids, and then pick out these peaks corresponding to single peptides and do a secondary fragmentation, breaking them into much smaller pieces to try to figure out from the masses of those smaller pieces what peptide actually contributed to a given peak. Again, the real thing is quite a bit messier than the ideal, but more or less we can figure out what peptides correspond to what peaks, and at least get some quantitation from this. This is much easier to automate, so there's a lot of hope that this is going to be the equivalent for proteins to what sequencing is for nucleic acids, but it is harder to automate still. The quantification is very iffy, and these problems of dynamic range are still an issue. If you have a sample with some things at very high abundance, you can't see the low abundance things. That's a hard problem people are working on, but basically it's widely considered the best product for a whole proteome high throughput method, although it's still really a research technology for looking at more than a few things. So that basically covers the technologies I want you guys to be aware of. I think we're about